Okay, in this video we're going to begin looking at BLAST and talking more about similarity and homology. So the first term we need to learn here is comparative annotation. Right here, comparative annotation. Now, comparative just means you're comparing sequences, and annotation is kind of like you're trying to find the parts. Like there are certain parts of a gene, introns and exons. If you have reviewed that type of structure, that's only going to be in eukaryotes. That won't be in bacteria, prokaryotes. But comparative annotation means you're going to look for the parts of a gene by comparing your unknown sequence to known genes. So comparing to known sequences in order to find the parts of an unknown sequence. So how do you measure the similarity? Now that's different than the comparative part, but the similarity is essentially, well, are the sequences the same in some way? What parts are, of course, similar, but what's happening with all the letters? So we've kind of talked about this some homology and similarity. But then how do you decide whether the two sequences are similar enough? Now there's where we haven't really gone in any depth. So we're going to begin doing that here in this lecture. Now next, we identify the functional elements. So these are the little pieces that do something inside a gene, and we want to find the functional elements, the smaller sequences inside the larger sequence that are known to have functions. So we may look at them and look at a larger sequence down here, but there's a small piece that can be identified as a known place where, say, a protein can bind, or that causes a particular shape in the DNA. The DNA is not just a straight, linear line of DNA. It bends and curves, and there may be a shape change in the structure of the DNA. Oops. If you can identify this particular sequence. And CFTR gene, here we're looking at a much larger, and we're saying, hey, this gene looks like the mouse gene or the dog gene, and we can compare that to it and find that. But it's just not a whole gene. It could be a shape, it could be a smaller sequence, it could be a whole gene, it could be a whole segment, it could be a large region, literally millions of base pairs across. And if you find certain trends like repeats, it's mainly made out of C's followed by G's, then we know that there are certain properties of that sequence, and we know that its shape has changed. But it may just be a gene. You compare the larger genome, maybe a chromosome, to a particular gene, and that tells you that it's probably CTRF. In other words, if it looks like the other three genes, is what this is saying. All right, so the functional sequences are what we are called negative selection. In other words, if it does something, now this is we assume that this is the case, that if something does something, and something, especially if it does something important inside the cell, that if it changes, it's going to kill the organism. So you're unlikely to see any changes in that sequence in any living organism, because if you change it, it dies. So they are under what we call negative selection. They don't change very often, because if they do, they die and they don't reproduce, so therefore you never see a particular change in nature. You just will not see this sequence. Maybe it has to do with a protein that makes up the ribosome. Ribosome is essential for all life. It's the little thing that does translation, and if you change it, it doesn't work, then nothing works. So you can't change a ribosomal gene very easily. You would be under negative selection, and therefore it has fewer mutations, because generally any mutation means it's not going to work. So more conservation and greater similarity. So you see that if things don't change between, say, a gene in a fly and a gene in a human and a gene in a mouse, if it's conserved, if there's a piece of DNA that doesn't serve there, it's going to be more similar, and we assume that that also leads to conservation. Now, this, of course, leads to a great deal of controversy when you go to try to figure out if these organisms are related. You might have two organisms that really need a particular protein to stay the same to survive, whereas another organism might have an extra copy of that gene, and it might not be as big a deal if one of them changes, and so you can't always measure similarity as a or use similarity as a measure of relatedness, though they try, and it's a complicated field when you get into that sort of thing. Now, the main way to measure similarity on the web, the way most people do it, is something called Blast, and it's based on the concept of similarity. Similarity without conservation, that's random chance. So in other words, if two things are similar, it might just be accidental, and that would mean that you might imply that it was conserved between these two organisms, but there was none. If you don't see that conservation across many organisms, say for the same gene, then it might just simply be by random chance. There is no guarantee that similarity leads to conservation. Now, without selective pressure, you might see it. It might be a slow mutation. Some regions of the genome change quicker than others for structural reasons, not functional ones. And then it might have recently diverged, like humans and chimps are more recent in their divergence than, say, humans and mice. Mice are much further apart. We diverged from the same organism many, many millions of years ago, and not just a couple of million. All right, something like that. So similarity and selective pressure, but sometimes they might have two distinct functions, but have the same selective pressures. So this is kind of harder to explain, but one, not all sequences have that are the same have the same function. You kind of always think that's the way it is. Well, this sequence does this, and this one, this sequence is identical, so you assume that sequence does the same thing. That's not always the case. Sometimes these things are structural, and they need to be there to make the region stable so that the DNA doesn't break. That would, of course, mean that it's selected to be there, because it isn't that it provided like a chemical function, but it provided a structural function, and that's not quite the same. So genes will have functional parts, but regions in between the genes might have structural functions, so it is kind of a function. But two sequences can also have more than one function. You might have a region of DNA that is both a gene, so it has a function, a chemical function, but maybe it also has a structural function. So there are cases of having both chemical and structural functions. I think that's a better way to explain it than the way they have in this slide. All right, we move on. How to measure similarity between sequences. So we need to, now that we kind of look at it, how do you measure? There's going to be some numbers, some minor statistics. So we have to always get a number that's kind of like, okay, somebody made a 70 and somebody made a 90. All right, and of course 90 is better. So we're going to do similarity, and we measure, like, okay, are these, like in a dot plot, you kind of move that measure around. You might have do I want my dots to appear whenever there's 70% sequence match, or maybe only 50% sequence match, or do I want it really high, 90%? And so that we have to figure out, well, how are we going to measure this similarity in the sequences? So similarity, mutations cause the similarity to decrease. So we want to measure something that will, or create a measure that reflects how mutations work, etc. And there are three types of mutations that we are going to worry about. And you should write about these when you talk about a sequence in a project or something, and you talk about, well, this one is different, this sequence is different from this sequence. You need to state it in ways like this. There are three types of change, mutation. You can have a substitution. In the first case is a substitution, so it started as ACG and it mutated to ATG. So you substituted a C for a T, so we call this substitution. When I changed the C to a T, it was substituted. You took one out and put another one in. Now the insertion of one base is basically adding one. All right, now you don't always just insert one. Sometimes you can insert many. It's going to be, might have a different name for it if you insert many, but a general insertion, as we call it, would just be the sequence had an extra base added in between two existing bases. So here ACG, and you're going to add an A in between the C and G. That's an insertion. A deletion, of course, is the reverse of that, where, okay, there was a C here and it got deleted, it got cut out, and now it's A followed by G. So these are your three basic forms of sequence change caused by mutation. Some are more likely than others. Some are more likely in certain places in the genome, like within a gene or outside a gene. You might get a higher rate of maybe insertions than you get elsewhere, or maybe you get substitutions. Substitutions may be the most common thing inside of a gene, but outside of a gene, that might be a very rare thing. That's actually not the case, but I'm just trying to make the point. Okay, next, here we have two sequences, S and T, and they diverged from a common ancestor U. So here we have a sequence that we've named U, and it diverged. It had some mutations, and so its descendants, this is the ancestor U, its descendants both had mutations in them, and we call it S and T here. So we start out with AGAGT, and what happened here is this a substitution? Well, AGA, there was a deletion here. We took a G out. Now over here, AGAG, we had a substitution. We turned a G into a C over here. So these two are related. All right, they have an ancestor here that that started with this sequence, but the descendants, the two children sequences, if you'd like to call it that way, are different from one another in that this one lost a G, and this one turned a G at a different place into a C. So, but they are similar. All right, they are similar in the sense that they came from this one, though it might be hard to tell that. And so we need to learn to measure or create a measure to record this change in a numerical way. Now, one way to do this is to draw alignments. Tree alignments are actually not common. This is not a common usage, but it is. You kind of get the idea of how it works by using it. So a dashed line where you had the deletion, there should be a base there and there's not. And over here you had the G, it used to be the C used to be a G, so it's a straight line. But this is actually not a common, a tree alignment is not a common thing. This is a more common one. This is how we would normally write an alignment. The A, the C, and G, you can see here, these are corresponding, and then the G, of course, you do use a dashed line where it would be. And so this is comparing S and T, the two children, the two descendants from the original. Now notice they did put a line between the C and G. That's to say correspondence. Normally you wouldn't put anything there. This would be blank because these are not the same letters. They're doing it this way just to show that that is the one that are supposed to be related. Normally in alignment that would be a blank because they're not the same letter. You typically only put the solid line where there is identical letters. All right. Here, at least they turned it to gray. So we score it. And of course, the deletion is actually a more serious change. So it's scored with a higher numerical value. So in this scoring scheme, and there can be many, if it's identical, we give it a +1. And if it's a substitution, you give it a -1. But if it is a deletion, it's a -2. So this is a more serious change. So you get a penalty for this. And so that's called a gap penalty. You get a mismatch penalty, as in these are not the same. And then if you get them the same, then you get a match bonus, which is here. So this is one way to create a score for our alignment. Now this is going to bring up the problems here that if not, how do you know that the ancestral sequence U, history of mutation? Hence, we get to the correct alignment. Well, this is not as easy as they make it sound. It's actually quite difficult to do this, and it's most of them that are done are, in my opinion, probably wrong, but at least you can get close, maybe. So yes, this is correct. You guess. You start out with these, and this is S and T, and you go like, wait a second, something's wrong here. Yeah, they're different, but it's not because there have been deletions or something. They really do align on top of each other. I have two C's here and two A's. I have two A's and two C's. They probably align, and that's just a simple substitution. That's the way to get to, actually it says maximize the score. Well, minimize the number of changes, and said these are not two completely different pieces that you've had all of these deletions here. No, these two align, the AA and the CC with a single letter in between. To me, that's obvious that that is a G that has changed to a T, or vice versa, or from some previous. So this would maximize it. This would minimize it. This would make the score lower. This one would make the score higher because you've you instead of having 10 changes. What you're saying here is that these were all deleted and these were all deleted. They'd all be -2. So it'd be like a -2 for each of these. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So it'd be a -20 score. And here, this one is +1, +2, +3, +4, -1. So it would be a +3. So this is +3 down here, and this is -20 down here. This is the more likely the correct one. Doesn't mean this could not have happened. So how do you know this if this happened or this happened? This is the most likely one, though. All right, how to decide whether two sequences are similar enough? Okay, this is a good question. Once they're aligned and you have a score, well, higher scores are better. And when is the score high enough to be evidence of conservation? So if you come up with a score, when is it high enough that you think that the two sequences may have come from a common ancestor? Well, we'll skip these symbology here. Can't say it today. We're going to skip some of this notation here. We don't need to know that the the sigma value and things like that here. Um, just know that those scores, once you have an alignment, um, they represent there's a probability associated with them, and you let the software do that. We're not going to calculate that. Um, but two sequences could be generated randomly. This is what you need to know. We're not going to do it mathematically. We're going to let the computer do it. But two sequences could be produced randomly here, as you see. All right, and they might be kind of similar. I see an AC and a G. If you look at it here, AC, and then skip something and there's a G. Here's an AC, some other letter, and a G. These could be related. You could move this sequence here down one so that this A corresponds here and the C corresponds. And then you could maybe say that there was an insertion of one of these letters and a substitution of the other one, and then the G. So you could align these. You would align it by moving the A to the right here, and that means this one and this one would align, and then you align this one and just assume that you'd have to insert an extra one up here. But they certainly could be, and randomly, look at that looks pretty good. But yeah, these were both randomly generated, and they do show some similarity. You just have to come up with a way to say how likely is this process. But we're going to let the computer do that for us. We're not going to do that by hand. There is a theory about how the alignment and the score should be at least a certain number, and we're going to look at that we're going to look at the score that we get, and we're going to go, okay, it's above or below a threshold. And if it's above or below that threshold, we say, yeah, those are probably come from the same ancestor. Or we're going to reject that and say, no, we don't think that's the case. These are just randomly happen to be similar, but not enough to say that they're related. And Blast can do this for us, and it uses an E-value. Now, ignore this here. There is an E-value. With this one, the larger number is better, but we're going to use this type of E-value where less than 1 is better. So for us, E-values that are reported from Blast, we're going to say they need to be less than 1, and in fact it needs to be like less than 1 to the -5th. In other words, 4, maybe 5 zeros, and then you start seeing numbers. So it's going to be 0.000001, and then you start getting interested in it. If it's 0.01, not too interested in it. If it's 0.5, no, not interested at all if it's 0.5. But if it's 0.00000005,738 or something, okay, that's interesting. That's enough zeros. You get 4 or 5 zeros in there, and we start getting interested in this sequence. So that's kind of how we will use E-values. You need at least to the -5th. Probably would like to have -6th or smaller, but if you get to the -5th, we'll say, okay, those sequences are similar enough for us to think they came from the same ancestor. And now realize this is for nucleic acids. When you start doing amino acids, you're not as picky. You can have larger numbers. In other words, less zeros in that 0.0000578 or whatever. But we don't need as many zeros when it's amino acids for us to be interested. But in nucleic acids, yeah, 10 to the -5th is what we're kind of looking for as a bare minimum in doing E-values for nucleic acids. And that's exactly it. So we want to be very skeptical. We get an E-value, and if E is greater than the 10 to the -5th, we reject it. So very skeptical. In other words, if it's greater. But once we get it equal or less than, if this value is here or less. In other words, 10 to the negative 6th is good, 7th is better, 8th is just wonderful. If you get negative 10th, they're pretty much identical. But e-values that are large, as in this number gets smaller, in other words 10 to the negative 2nd, that's not good enough. That e-value is too big. We would reject that. So just keep in mind how this works. Comparative annotation. Blast is going to compare two sequences, and it's going to come back and it's going to align them. If it's just two sequences, it can align more, but it's going to align them and it's going to give you an e-value. And if the e-values are well, actually low enough, then you would, it says high enough, but actually we're looking at the smaller numbers as being better. So if you get a good enough value in an e-value, you're going to go, oh, these two are related for sure. And I'll show you how to do that in a second. All right.